to have you. Apparently we're recording. It's a pleasure to have you here today, and we will start off with Jane giving a little bit of a background about our RadNet City of London. Over to you, Jane.
Hi, good afternoon. Thank you for joining us for this. It's our first seminar within the RadNet City of London seminar series that has featured molecular radiotherapy. So we're very excited to have a different form of radiotherapy being talked about today. And we have with us Jean-Pierre Pouget, whom Sam will introduce in a few moments. Just a few housekeeping things. Please mute your microphones and turn your cameras off. And note that you should post your questions or comments to the speakers in the meeting chat, or raise your hands, and these will be answered in the Q&As. And also that the meeting will be recorded. So when we conceived this talk and having a talk about molecular radiotherapy, sorry, I'm struggling to change slides. There we go. We had been working within the UK community to create a network for molecular radiotherapy, simply because we lack the raw materials sometimes to do this type of research. So, of course, molecular radiotherapy is the injection of radioactive substances for therapeutic effect. And if you don't have those radioactive substances, the actual radionuclide raw material, then this really hampers research. So I just wanted to highlight here that we have been working with people such as the National Nuclear Laboratory, who are going to be supplying us with radionuclides from nuclear waste, Mithras from King's, which is a radiochemistry program. We have an advocacy program through Queen Mary University of London and the EPSLC called Radionuclides for Health UK. And this is to really persuade the government that they need to invest in the infrastructure that we need in order to be able to produce radionuclides in this country, use them, radio-label them, administer them to patients in clinical trials. And this is what we're trying to do within RadNet City of London, which we have in RadNet City of London. There is a program on molecular radiotherapy. So thank you very much for listening to that, and over to you, Sam.
Thanks, Jane. I would also just like to highlight that we're really excited that these seminar series are basically a chance for us to kind of connect with each other, both within the RadNet units in the UK, but equally with external speakers, just like Dr. Jean-Pierre Pouget. Now, to introduce Jean-Pierre would take quite a lengthy amount of time in terms of all the things that he has been contributing in terms of the radiobiological studies in molecular radionuclide therapies. In short, though, in 2000, he obtained his PhD from the Curie Institute in Paris, went on to do postdocs at Barts here in London, and then also moved back to Paris shortly after that to work for the French Radiation Protection and Nuclear Safety Agency. Now, most recently, and I guess this is where we know his work most from at the moment, is his move to Montpellier, where currently he is the research director and the leader of the radiobiology for targeted and personalised radiation therapy. And he also works at the Cancer Research Institute of Montpellier. Now, when it comes to his accomplishments in terms of the research that he carries out, I guess it's safe to say that his remit is all about creating new radiopharmaceuticals for imaging and targeting cancer, and to be able to do this in a way that is translationable to the clinic. So in a way to actually make an impact to real life patients. He doesn't seem to limit himself to the type of radionuclide. He's worked with alpha, betas, and Auger electron emitters, as well as with gamma and X-rays. Now, of course, personally, I'm very excited about the Auger aspects of his work. And equally, he likes to explore the different mechanisms in which molecular radionuclide therapy can be effective. So not just in the targeted radiation realm, but also through indirect and bystander responses, as well as through signaling pathways. Now, having published more than 70 papers, book chapters, etc., and been successful on the patenting side as well, you know, we can't expect him to explain everything in the next 45 minutes. So instead, I believe his focus today is going to be on looking at the role of vesicles released by cells in the therapeutic efficacy of molecular radionuclide therapies. So thank you very much, Jean-Pierre. I look forward to your talk. And as Jane said, if there are any questions, do already post them in the chat and we will be going through them at the end of the talk. Thank you. Bye.
Good afternoon, everybody. Thank you very much, Jane and Samantha, for inviting me, and thank you also RadNet City of London London Network. Then today, as mentioned, I'll try to move that. My talk will be about the role of extracellular vesicles, and I will talk also a bit about DNA fragments in the non-targeted effects of molecular radiotherapy. I will talk more about targeted radionuclide therapy, but it's the same. Just to remind you, about 50% of cancers are treated by external radiotherapy that use X-rays radiation. But external beam radiotherapy is mostly dedicated to localized tumors, and it's not possible to use this approach when tumors are metastatic or diffuse. And that's where the field of targeted radionuclide therapy takes place, because it consists, as mentioned by Jane, of injecting radiopharmaceuticals. It means radiolabeled compounds in the body that will specifically target cancer cells and specifically irradiate them. Then there are quite spectacular success of TRT. One of the most striking, as you can see on the slide, is a patient with prostate cancer that has been treated with PSMA radiolabel with an alpha particle, and you can see the monitoring of the disease following several injections. And several specialties have been approved, and one in particular in this type of cancer based on PSMA. Then that's a very attractive and developing field. Just a short introduction about the differences between TRT and conventional radiotherapy. As I said, conventional radiotherapy use X-rays, but which have, of course, pass in the matter. With particles, we have a finite path in the matter, and we are mainly three types of particles emitters that can be used in TRT: the beta, the alpha, and the Auger electrons. And the range can be from several nanometers to several millimeters. But also one of the main difference with X-rays is it's that using TRT we are able to use what we call ILET radiation. It means this in particular alpha particles and Auger electrons that will produce dense ionization and will produce complex damage in cells. And this is not observed with X-rays. Then what do we know about the way radiation acts in biological samples? What we know, it's the paradigm of radiobiology, is that only the cells traversed by particle can be killed. And in this context, DNA and DNA double-strand breaks constitute the main lesions produced by radiation. We also know, I don't know if I can have the pointer, maybe not. We also know that the biological effects, as observed during external beam radiotherapy, are proportional to the dose, radiation dose. And luckily, the toxic effect occurs at a higher dose, which allows to have a therapeutic window where we can treat patients. Okay, then a few years ago, we started a quite interesting study because we use radiolabeled antibody. I'm sure that some of you must know this story. Then we targeted cancer cells. We targeted the carcinomatic antigen with radiolabeled antibody. And the particularity of this system is that when the antibody binds to the cell, it remains at the cell surface. The receptor is not internalized, and we have a non-internalizing antibody. And we were quite surprised. Here it's a survival of mice, it's a Kaplan-Meier graph. We were quite surprised to see in blue that we have some tumor growth delay, because as I mentioned, the Auger electrons are very short range. When they are located at the cell membrane, we were not expecting them to have any effect on the DNA. Then that was the first observation, and you can see here that we were treating quite small tumors. It was 1 to 2 millimeter peritoneal carcinomatosis, but these results were quite surprising. And this observation was confirmed in several in vitro systems, and you can see here again that the cell membrane, for example, the cell membrane, when the antibody was located at the cell membrane, it was more cytotoxic than when located in the cytoplasm. But of course, if we managed to bring the radioactivity in the nucleus, we were more efficient. This was confirmed by dose assessment. We showed that when the antibody remains at the cell surface, when it's bound to an Auger electron emitter, most of the dose is deposited at the cell surface. What we knew from the effects of radiation of cell membrane came from external beam radiotherapy. And just to remind you, the cell membrane is a phospholipid bilayer. It's made of sphingolipids, cholesterol, proteins, phospholipids, and we were more interested in one of these lipids, the sphingolipids, which is made of a fatty acid and of sphingosine. And it was shown from external beam radiotherapy that when we irradiate the cell membrane, we could oxidize the polyunsaturated fatty acid constituting these lipids. And it was also possible to activate some cell surface receptors or activate channel ion. But it was also possible to observe, that ceramide-enriched lipid raft domains could be formed. Then external beam radiotherapy, there were several studies now, it's more than 20 years ago, that showed that when cells were irradiated, the acidic sphingomyelinase enzyme, which is located close to the cell membrane, could be activated by hydroxyl radical. And once activated, it could hydrolyze the sphingomyelin, which is part of the sphingolipids, and into ceramide. And ceramide, as shown here, which is another lipid, could participate to lipid raft formation. These lipid raft domains are present in all the cells, but when we radiate, they will tend to gather all together and form large platforms at the cell surface. They are, because of chemical reason, because of melting point, they are more rigid than the rest of the cell membrane. Then they are floating domains at the cell surface, and they tend to activate receptors. And it was shown that they could participate to a cell death mechanism. Then we were just thinking, okay, when we use radiolabile antibodies, they, in all the cases, but it's even more pronounced with Auger electron emitter, they will irradiate specifically the cell membrane. Then maybe that's why we have some effects with a non-internalizing antibody. I will just show you some results with Auger, but we had the same, and they have been published with alpha particle emitters. Then we showed that effectively in cells exposed to Auger TRT, the acidic sphingomyelinase enzyme was activated. Then we are here, and we show that this led to the production of ceramide, as shown here by flow cytometry. This led to the formation of lipid raft in green here. Then you can see that in the irradiated cells, all the cell membrane is green, indicating the formation of these domains. And we show that if we were inhibiting the enzyme, the acidic sphingomyelinase enzyme, using imipramine, we could restore the survival. Then in black, you have the efficacy cytotoxic effect of TRT on cells. But when we incubate cells with imipramine, we lose this cytotoxic effect. Then we said, okay, we know now that lipid raft are formed at the cell surface. Then what happens if we disrupt the lipid raft? And here we use two drugs, the Philippine and the methyl beta cyclodextrin, that are known to disrupt lipid raft. And you have the clonogenic survival of cells exposed to TRT. And when we use the combination TRT and Philippine, we restored part of the survival, and the same was observed with MBCD. Then these results indicated that lipid rafts are involved in the therapeutic efficacy of Auger emitters. Okay, then just to summarize this first part, we show that Auger TRT could kill tumor cells, which was quite by targeting the cell membrane, which was quite unexpected, and we showed that lipid rafts were involved in this mechanism. Then our view of the radiobiology of TRT evolved from a DNA-centered view that I mentioned at the beginning of my talk, and which exists, of course, and play a crucial role. And we moved to this view where the cell membrane, but I didn't show also some result, but other subcellular compartment could be involved, and all these subcellular compartments, of course, communicate. But we are moving from the target cell theory view to something more integrated. We also observed something quite interesting. Usually when we irradiate using external X-rays, most of the biological effects, of course, it depends on the biological effects, but for example, the survival or the induction of DNA damage were proportional to the dose. And usually then the biological effect increase when the dose increase. In our case, in most of the situation, we didn't observe such increase, and we always reach a plateau, as like shown here. And if you look at the results I showed you, we observed this type of plateau. Then, when the activity incubated with cells increase, we don't kill more cells. Then this was quite unexpected. But it was also observed to some extent in patients. Here it's patients treated with Lutathera. The patient 1, the patient 2, they received the same absorbed dose, 48 and 47 gray. And you can see that the initial tumor was much bigger here, but at the end they respond differently. And the patient 1 responded much better than the patient 2. And of course, there are other reasons, but as compared with external beam, I would say that it's really something showing that, in particular, in our case of TRT, it's difficult to correlate biological effects with the absorbed dose, even if sometimes there are, of course, some correlation. And external radiotherapy showed that this lack of dose effect relationship, when occurring, could be related to bystander cytotoxic effect. Then, what are the bystander? I'm sure that you all know about that. In the field of radiation, we call targeted effect the biological effects that are observed in cells that are effectively hit by radiation. Then here, I hope that you can see my mouse. You have this type of situation where the DNA is damaged by radiation, but in fact, you consider the whole cell irradiated. And we call bystander effects biological effects that are due to intercellular communication and that are observed in cells that have not been irradiated. It means that these cells have received a dose of 0 gray. Then we were wondering whether bystander effects could be responsible for the type of dose response that we observed with Auger TRT, and if lipid raft could play a role in this bystander effect. And we use a protocol for preparing conditioned medium, and that's something quite classical. Then we incubate cells with radiolabel antibody. After, in our case, after a given time, we remove the radioactivity, we rush the cells to remove unbound radioactivity. We incubate cells with fresh medium, and we collect this medium to transfer it to what we call recipient cells. Then this conditioned medium will allow to measure bystander effect in these recipient cells. These recipient cells have never been in contact with any radioactivity. They are just in contact with the conditioned medium. Then the idea is that soluble factors or factors released by irradiated cells will be present in the medium and will produce some biological effects. Then we can carry on with the cells that have been, we keep the cells that have been exposed to radiolabel compound, and we call them donor cells. And these cells have effectively been irradiated, and we can measure in these cells the direct effect of radiation, the targeted effect of radiation. Then we use these protocols in cells in our models. And here you can see that when we expose cells to TRT, we have this type of activity-related relationship. And here you have the same results obtained for bystander cells, the recipient cells. And you can see that the dose response is not so different. And here the cells have just been in contact with conditioned medium. When we treated donor cells with Philippine, as I mentioned before, we restored the survival of the donor cells, but also of the recipient cells. And this was also observed with MBCD. And we confirmed that this was also related to DNA damage measured in donor cells here in the upper panel, which we worked with 2 antibodies in this case. You can see the damage in the recipient cells, but when we use Philippine in donor cells, we reduce the level of damage in recipient cells. Then that indicates that bystander genotoxic effects are produced during OG-TRT. And they can be abolished by Philippine and MBCD that I didn't show. We investigated, I won't go deep in the details, but we investigated what could be the signaling pathways involved. And using protein kinase array, we identified in red all the proteins that were overexpressed. And you can see that our unlabeled antibody had no biological activity. And when we use Philippine, we abolish most of these signals. We, in fact, confirmed that P38 and Jun kinase signaling pathways were involved, as shown here. And here we use some inhibitors to show the involvement of the signaling pathway. Then for P38 and Jun kinase, and we show that we confirmed the involvement of this signaling pathway. Okay, then we wondered, well, in vivo, we could also observe this type of bystander effect. Then we treated mice bearing carcinomatosis. It's always our model, as shown here with SPECT-CT imaging, the same type of tumors as mentioned before. And mice were injected with TRT twice. It was OG-TRT in this model, but we also did that with alpha particle emitters, and we combine it with MBCD, which is, as mentioned, a lipid-wrapped disruptor, or with pravastatin, as it can reduce the cholesterol level. And what we observed is that TRT was less. Here you have in red the efficacy of TRT, it's average tumor weight. Then TRT was less efficient, not anymore statistically different from the control when we combine it with MBCD. And the same results were obtained with pravastatin, then indicating that pravastatin, or drugs that traps cholesterol or modify its metabolism, will affect the TRT efficacy. And I just added that before the talk. Here you can see the effect on DNA damage at the tumor level. When we injected mice with TRT plus MBCD, we observed a lower average level of DNA damage in the tumors. We also had a look at what was the spatial distribution of DNA damage in these tumors. And this is autoradiography, which has been performed on slices from tumors. And we also use the following slice just to measure DNA damage by an immunohistochemistry approach. And you can see here it's 53 BP1 detection. And then you can see that in our model in red, it's the distribution of the radioactivity at the periphery of the tumor that's really due to the antibody we used, which was mouse antibody in a human tumor. Then we had a very heterogeneous distribution of radioactivity, but this was very helpful because we had some areas in the tumor that received 0 gray. We did some voxel dosimetry on different slices, and some areas didn't receive any radioactivity. And with a conventional approach, we would expect DNA damage mostly be produced where the radioactivity is, where the absorbed dose is, then at the periphery of the tumor in our case, in some spots. But in fact, what we observe is that it's a blue curve in this type of model. We observed that the level of DNA damage was similar all over the tumor. That indicates that some factors migrate from the most irradiated areas to the area that receives 0 or very low dose to produce DNA damage. And finally, then, as mentioned, the level of DNA damage was roughly similar all over the tumor. And that's an indication of what we call the bystander effect. It means that DNA lesions are produced beyond the range of particles in non-irradiated area. Okay, then just a summary of what we showed. This has been published, and if you want to have a look at it more in detail, you will find it. Then we show for both OG and alpha TRT that when the antibody binds to the cell membrane, it will lead to the formation of lipid raft, activation of P38 and Jun kinase signaling pathways. Of course, and in particular in the case of alpha particles, we will observe also DNA damage due to direct effect of radiation because of the range of particles. And this finally will gather in common signaling pathway, leading to oxidative stress that will in turn damage again DNA and so on. And we assess that depending on the model, the contribution of bystander effect was always between 30 and 35%. When you kill 3 cells using TRT, one will be killed by bystander effect and the others will be killed by direct effect of radiation, which is quite a lot. Okay, then we looked at what could be the possible factors involved in this bystander effect, and finally, we investigate several fields, and finally, we focus on the extracellular vesicles and wondered whether they could be involved. Okay, then our question was, are the extracellular vesicles released by irradiated cells, can they participate to this bystander effect? Just to remind you about the extracellular vesicles, they are small vesicles released by cells, and they are part of, and in this project, we were in what we call exosomes, which are the smallest extracellular vesicles. They have a size between 50 and 100 nanometers. But usually extracellular vesicles also include apoptotic bodies, which are the largest, and the microvesicles, we have intermediary size. And exosomes are formed from endosome and from multivesicular bodies within the cells, and they contain many types of substances, RNA, microRNA, DNA, proteins, and different types of molecules. And the composition depends on the type of cells, on the phenotype, and so on. Then it's a huge, very wide field. We use a conventional protocol for isolating exosomes from cells treated with TRT or with X-rays for comparison. And here is the protocol that you can find in the paper, but it's a very classical protocol. We end with a sucrose gradient, and we isolate and characterize exosomes. They should move, they don't move, but it doesn't matter. And that's we use a nanocyte nanoparticle tracking analyzing nanocyte system to characterize their size. We confirm their nature using Western blot, and we next could work with these exosomes. Then the first thing that we did is that I cannot see my title, but I know my slide fully. First, we measured the bystander effects in cells. It was melanoma cells exposed to X-rays, and that's the upper panel. Then you can see that with X-rays, with cells exposed at 0.5 gray, which is not a lot, we have some cytotoxic effect, but no bystander effect here as measured in the recipient cells. When we expose the B16F10 cells to exosome collected from these irradiated cells, we didn't observe any significant decrease. When we did the same experiments with OG-TRT, then in black, you know this curve because I showed it several times, then the cells were killed by OG-TRT, but we had also a strong contribution of bystander effect as shown in recipient cells. But when we collected the exosomes from these cells and when we incubated with B16F10 cells, we also observed that the exosomes were cytotoxic. Then it means that the cytotoxicity and the nature of exosomes will depend on the radiation type, but with OGTRT, they were quite cytotoxic. We investigate how they could kill the cells, and we look at apoptosis, and at the end, we didn't observe any significant induction of apoptosis, even if there was a trend, but we observed a significant increase in DNA damage in the cells exposed to exosome, and also of micronuclei. Then that showed that these exosomes were highly damaging the cells, and that could explain what we observed, this cytotoxic effect. Because we were interested in ceramide, as I mentioned before, and because it was described in the literature that exosomes could contain ceramide, we quantified the ceramide in exosome, and we found that during OGTRT, the exosome collected from cells exposed to OGTRT had a high level of ceramide here, and when the cells were incubated with imipramine, the exosome didn't contain any ceramide. We showed that the exosome from OGTRT, we showed that their cytotoxic effect could be explained partly by the presence of ceramide, as shown here. When the cells were exposed to TRT combined with imipramine, we collected the exosomes, and these exosomes incubate with cells, didn't kill the cells in blue here. Okay, oops, sorry. We next had a look at the content in DNA, because it's well known that exosomes contain nucleic acids, and we showed that exosome for OGTRT also contain dsDNA, in addition of ceramide and so on, and proteins. And this was quite interesting for us because it was described in the literature that when exosomes contain dsDNA, they can activate immune cells, and this is shown here. The cancer cells release exosome containing DNA, and this can in turn activate SIGA-STING signaling pathways, as shown here, leading to phosphorylation of ERF3 that will in turn produce interferon gamma and participate to priming of immune response. That's observed here, but the DNA damage can release, for example, in the cytoplasm, can also activate these signaling pathways in the tumor cells. Then our view of TRT changed, our radiobiological view, because we started from a DNA-centered view, we moved to a more wall-centered view, and all this is part of the targeted effects, and we showed that bystander effects could be involved, then in cells that have not been irradiated, and now we are interested in the systemic response, as some factors, and exosome in particular, can participate to the activation of immune system. Then first we knew that TRT was more efficient in immunocompetent mice than in mice atemic mice, then indicating that T cells, mostly T cells, participates to TRT efficacy, and this was true for the 3 types of TRT. More interestingly, we showed that when we collected exosome from cells exposed to TRT in vitro, these exosomes were then collected, and we injected them intratumorally in mice. We performed 4 injections, and as you can see here, this led to a significant tumor growth delay as compared with the exosome collected from non-treated cells. And this was observed in immunocompetent mice, but not in nude mice. This indicates that in vivo, these exosomes participate to the therapeutic efficacy of TRT, but this is mediated mostly by immune system. Okay, the Kaplan-Meier, but we were quite surprised because so far we were thinking that DNA content in exosomes could will be responsible for most of the biological effects observed. And in fact, as shown here, that corresponds to the 4 injections that we perform in mice. And don't ask me why, but the results were gathered like that. But the conclusion is that here in blue, it's the control exosomes, then from non-treated cells, and the red exosomes collected from cells exposed to TRT before the injection to mice. And finally, the amount of DNA in the exosome was not significantly different between the treated cells and the non-treated cells. Then it means that here, the biological effect that we observed in mice, the therapeutic efficacy or tumor growth delay, could not be strictly related to the amount of DNA contained in exosomes. Sorry. Then, okay, these studies are still ongoing, but we wondered whether exosome then could activate immune cells, and these are preliminary results that we need to confirm. But here you have some, in fact, we use CRISPR B16, okay, it's B16 F10 cells that are CRISPR and negative for CGAS, KO negative for CGAS, or KO for STING, sorry, KO for CGAS or KO for STING. And what we showed that, in fact, we showed that we incubate these cells with, that was macrophages. We showed that when we incubate these cells with exosome collected, when we incubate these cells with exosome collected from these cells, they were not activated in the cells that were lacking CGAS. Okay, but when we incubated these cells with exosome lacking STING, we could observe an activation. Then this activation is ongoing. The study of this activation is ongoing. Then with some RT-qPCR approach, and also we have some KO model, THP1 and for CGAS and STING KO. Then we want to check whether the extracellular vesicle will be able to activate or not. And this will indicative of what the extracellular vesicles will carry. Okay, what was surprising, then we knew that the extracellular vesicle doesn't need CGAS in immune cells to activate the cells. What was surprising is that when we look at tumors, here the B16 cells are deficient in CGAS, and you can see that the TRT, that was a revolution, the TRT was not efficient. In the STING, TRT was efficient, then indicating that even if the cells, I don't know if I have a conclusion, yes, even if the cells are deficient for, the tumor cells are deficient for CGAS, when the cells are deficient for CGAS, TRT doesn't work, but if they are deficient in STING, the TRT works. Okay, then we go back to the tumor cells, and we effectively show that in cells exposed to TRT, here it's OG-TRT, we could detect cytosolic DNA that accumulated in the cytoplasm of these cells. And as I showed before, we knew that this DNA in cytoplasm could activate CGAS-STING signaling pathway. Here you have the amount of DNA as a function of time. I won't go deep in the details, except if you have some question, but you can see that it increases and next it decreases after incubation with TRT. Okay, then we are here. Then we show that there is cytosolic DNA. We show that CGAS is important in TRT response in tumor cells. STING is not so important, which was a surprise because we knew that STING would participate, as I mentioned before, to interferon production and through activation of ER3. And we show here in Western blots, they are not quantified, but we show that they are not quantified on this slide, but we show that in the case of OJ-TRT, cGAS was effectively activated in this type of cells. We had also a transient activation of phosphosting and also some effects on phosphor here. Oh, sorry, here. Okay. The role of cGAS in tumor cells was quite interesting because we showed this was done in collaboration. And we showed that using TCGA data, we showed in patients that had been treated for melanoma, skin cutaneous melanoma, they had been treated only by radiotherapy, and we showed that the response of this patient was better when cGAS expression was high. Then that confirms what we observed in the case, maybe not, but that correlate with what we observed in TRT, that cGAS is crucial in cancer cells for successful TRT. Okay. And we also observed that sting was not so important as shown here. And on the right, sorry, I didn't mention that for surgery. Okay, then here it was the same patient. Sting expression in cancer cells was not prognostic of the success of radiotherapy. Okay, I think it's my last slide. Something that we observed, in fact, because we said some extracellular vesicle can be carried from cancer cells to immune cells. We observed that in cGAS cow mice, that TRT was not efficient. Then it means that if the immune system, in particular of mice, is cGAS cow, we won't have any modification of TRT response. But when sting is knockout, we have a loss. Sorry, we have a loss of TRT efficacy. Then that also correlates a bit the results we obtain in our in vitro experiments, showing that the extracellular vesicles maybe they carry the sDNA, but maybe it's not most important, and other factors like cGAM could be participate to TRT efficacy, and this has been shown in literature in other contexts, but that's something we are working on now. Okay, then just to summarize my talk, that's the general overview of TRT. We show that bystander cells and systemic response are involved in TRT. Then we are not considering only the DNA-centered view of TRT. We show that cell membrane was involved. It's not the only subcomponent, but it's involved through ceramide-enriched domains. We show that cGAS sting is activated in cancer cells. It's not required in immune cells, as shown on my last slide. We show that sting is required in immune cells, but not in cancer cells. And then now we are investigating how we can explain the efficacy of extracellular vesicle injection in mice, considering the content and with a special focus on DNA and cGAM, and also some parameters dealing the nature of extracellular vesicles and of course the nature of radiation. I would like to thank my collaborators, my team in Montpellier. Most of the results I showed today were from Salome Payas, Riyad Lajunlu, and the most recent unpublished results from Jihad Karam, with PhD student, and Julie Constanzo, who is a research scientist of my team. I would like to thank all my collaborators and you for your attention. Thank you.
Thank you very much, Jean-Pierre, for that quite succinct story, and it's especially nice to see it all put together into this new model of yours that was just recently published. I would again like to invite people to submit questions either in the chat or feel free to put your hand up. To get things going, however, I was wondering, Jean-Pierre, if you could perhaps share your insight into the Auger electron emitters that you work with. And, you know, although you're showing that you can bypass one of the main difficulties when using Augers, which is that you don't need to target the DNA, you can target the cell membrane, for example. What about the difficulties when it comes to the high photon electron ratios that you get for these Auger emitters that stops the translation to the clinic?
Yes, yeah, it's a big debate in the Auger field. Yeah, it's we all want to target the Auger electron emitter in the DNA. We know that it's not so easy because usually we use radiopharmaceutical that are most of the time degraded in the lysosome of cells after internalization. Then that's the first point. And okay, for targeting, then just to cell membrane is efficient, but of course it's less efficient than bringing the Auger in the DNA molecule. We are also less efficient if we are just in the nucleus than in the DNA, and so then okay, then cell membrane is an option. After that, about the choice, if I understood well your question, Samantha, the choice of Auger, yes, it's very difficult to find a good Auger electron emitter. Then what is a good Auger electron emitter? Probably everybody has his own definition, but Auger electrons are, I would say that they are efficient, they are highly efficient if you don't have any other radiation that will generate side effects and bone marrow toxicity or whatever. By themselves, I would say that except the situation where they are really in the DNA, they are not powerful emitters like alpha particles, but the interest of using Auger electron emitters that they are pure emitter, they don't produce side effects, they don't generate radiation protection concern. And the difficulty today to have a nice Auger electron emitter is to have something producing enough Auger electron per decay, but also not energetic gamma rays like with indium. And I worked with indium. Many people, of course, work with indium, but indium is, it's penalizing because they are very energetic gamma rays that generate radiation protection concern. And the efficacy, in addition, of indium, it's a very low Auger electron emitter. It depends on the physicist calculation, but yeah, it's 7, 8 Auger electrons per decay, which is probably not enough. Maybe an isotope with such number of Auger electron, but without any side radiation, energetic radiation would be good. I don't know if I was too long and clear, but
you were lengthy, but no, it was clear. Thank you very much. We also have a question from Alec. Alec, would you mind turning your video on and unmuting yourself for your question?
I see, yeah, you sent a request. Thank you. Let me just turn my hand off. Yeah, thank you very much for the talk, Jean-Pierre. The work from you and your team is it's a really interesting story. So I just wondered, with your models, your in vitro models of the direct and bystander effect, where you wash the donor cells and then you're obviously taking conditioned media off. So I wondered, with the donor cells, if you leave that conditioned media on, is there an enhanced effect? And I guess the reason I'm asking that is, do you think that there is actually the factors in that conditioned media, these bystander factors, they have a paracrine autocrine effect? Are they important in the kind of the direct effect as well?
Yeah, no, it's a very good question, because when we started, we say, okay, we will measure, of course, bystander effect in recipient cells, but you're totally right, because it depends on how long you incubate your donor cells with the radiopharmaceutical. But in our case, we incubated 48 hours, which is a lot. And during 40, we had, when we consider the determination of direct versus bystander, we had to consider that in what we measured as direct effect, there was a contribution of bystander effect because of paracrine and autocrine. Then we have, it's not a subtraction, it's more, but it's not complicated, but you need to use all the formulas. We use a Bliss formula that which is used for every time you combine drugs or two effects, to discriminate between what is due to direct and what is due to bystander effect, even in donor cells. In recipient cells, it was easy because you determine the bystander effect and you use this value to take it into account in the donor effect. Yeah.
Okay, yeah, thank you very much.
Yeah, thank you.
Thanks, Alec and JP. We also have a question in the chat from JP, which I'm guessing isn't yourself, Jean-Pierre, which is, do you think that the bystander effects actually induce senescence?
It's a good question. I would say we have never measured, but I would say, yeah, it's probable. Yeah, it's probable. I think it's likely. Sorry. Yeah, we have never measured senescence, but yeah, it's something that yes, was to be considered. Yes. Yeah. I've never measured, but we have some project where we're interested in that. But yes, I would say yes, probably. But it depends on the on the stress. But yeah, we need something that needs to be measured.
Yes, Jane.
Hi, Jean-Pierre, thanks so much for such an interesting talk. I was really interested in your exosome work, which seems like really exciting. I just wondered about the DNA. You know, you said you got the same amount of DNA in your non-radiated as your irradiated. Have you actually looked at the difference between the DNA and the exosomes? Is there any difference? Or?
Yeah, it's a good question. Yeah, I didn't show all the results because we need to work more on that. Initially, the idea was, okay, with what we know is that the activation, for example, of CGAS signaling pathway depends on the length of the DNA. And initially we say, okay, when we use alpha particles, we will have complex damage and probably DNA will be different. Probably the length will be different, but the quality will be different. And what is difficult is that, for example, with Auger, we showed that the amount of DNA was higher than in control, and we had some efficacy of these exosomes when we injected them in mice, but for example, with X-rays, we have more DNA in exosomes, but the therapeutic efficacy is not better. Then really it's the question of quality of the DNA. And we are doing some sequencing and trying to characterize this DNA in exosome. Yes, I think it will depend on the type of radiation, the quality, and the amount is a parameter, but it's not the only parameter. The size is has been described to be also a key parameter.
Thank you.
If I may, I'd like to ask another question, which is that, do you think that we're going to be moving in molecular radionuclide therapy based on your results so far in the next 20 years? Do you think that we'll move from molecular radiotherapy as we know it now to just basically irradiating or using extracellular vesicles from cells that have been irradiated with radioactivity and injected?
It's something we are working on. Because I mean, it's something quite exciting but the field of exosome is huge. I mean, and there are in the field of radiation therapy, and I would say it's worse in the field of TRT because we are not so many teams. But in fact, when you start looking at exosome and you discover that people have been working for, I don't want to exaggerate, but maybe for 10 years or more than 10 years on exosome, they try to target them to tumor cells. They are already, how can I say that, generator of ex vivo. As you said, there are companies that they have reactors to produce exosomes and so on. Then, I think it's a good idea, Samantha. I had the same, but I think it's maybe too late not too late, but people already thought about that. And it's a bit depressing to see that, yes, they already thought about that. And there are companies selling exosomes, and I don't know everything about what they do, but yeah.
It's not so much that I'm concerned that my amazing idea or thought is already being used, but more that am I making the work that I'm planning on doing in the next 10 years obsolete?
Yeah.
Bypass.
Yeah, I think if we show that it's really efficient because it was intratumoral injection, I think it would be nice, because it means that you inject less radioactivity, and yes,
you overcome a lot of the issues.
I guess, yeah, it would be a contribution. Maybe it should be a combination. I don't know, but it's a huge field. It's a huge field.
Alec, back to you.
Hello again. I'm back for one more question. Something just straight on from what you said, Samantha, there. So I just wondered, with the exosomes that you've kind of been looking at, are you finding that tumor cells are particularly sensitive to them, or are they just toxic to any cell?
It's a good question. I don't know yet, but it's something we need to consider. Yeah, it's we know that okay, sometimes exosomes carry receptors that make also that they can have different effects on different cells. But we try to cross between melanoma, pancreatic tumor cells, or exosome from generated in cells exposed to Lutathera, and to have a look on. It's not so clear for me. We know that it doesn't always cross react. Then sometimes it doesn't work. Maybe it's related to receptors, the way they are catched by the recipient cells. I don't know, but it doesn't always work when you cross. Yeah.
Thank you.
Yeah.
I guess just a final comment in the chat from JP, which apparently stands for Juan Pedro, which is quite lovely to see.
It's not me.
Jean-Pierre, who just had a comment, I guess, about senolytics, and that perhaps that combined with TRT might improve the efficacy of them. I don't know if this is something that you're
about. Sorry,
senolytics. So eliminating senescent cells.
Okay, I'm not competent. I'm reading the new. Yes. No, I'm not competent enough.
A good point, Juan, Pedro, in that case. What I might end with now then is Paul's final question. Obviously starts with the compliment of great talk, which we would all agree on. He then goes on to say, how might we go about quantifying the extent of non-DNA targeted effects that are actually important in the clinic? For example, if a tumor shrinks by 50%, can we work out how much of that is direct versus indirect?
Yeah, it's a very good question. It's a bit provocative now, but it's totally true. It's a bit like when people combine immunotherapy with or when they talk about abscopal effect in radiation therapy. There are cases reported for abscopal effect, but it's not so frequent. And but sometimes it's just when you use indirect way. For example, it's the same for immunotherapy. People use immune checkpoint inhibitor, and they show that it works better. Then it means that this was contributing to the response. I think it will be more this way. If we find, for example, our model with the cholesterol, of course it's in preclinical, but if it was demonstrated in patient when you modulate the level of cholesterol, the treatment, the people who are treated against cholesterol, if you were showing that you could modulate the efficacy, but of course there are so many other parameters that it's difficult, but I think it's the only way to show that this type of non-targeted effect can contribute. What is possible is as I showed in the tumor slices in mice. It's a good, I think it's not, we are not the first one doing that, but it's a good way to show when you measure damage where cells have not been irradiated, when you have no radioactivity, it means that even in mice, you have bystander effect. And there were also people, and I will end with that. Now it's maybe more than 10 years ago, they used microbeam irradiation, and even in mice, and they were irradiating just small area of the tumor, and they were showing that there were damage everywhere. But it's not, there were no direct evidence, I guess, for showing this.
Yeah, nor are there any real ways of determining one from the other in the clinic at the moment, I guess. That, I guess, I'm going to end the session there. Juan Pedro has sent you a URL to basically a link to Senolytics, so thank you very much for that. And I'd like to thank everyone for attending, for the great discussion after the talk, JP for sticking to time, and I wish you all well. Thank you again for the talk, and until the next time.
Thank you for the invitation, and thank you everybody.
Bye all.
Yeah,
Bye.
thank you everybody.
Thank you.